Happy morning, guys. Today we shall see about the structure of proteins, right? So already we know that this is just a protein amino acid formula, right? This is an amino acid and this is an amino acid, right? So here an amine group is here, here an acid group is here. So both of them are amino acids. When these amino acids combine together, they join together through a linkage called peptide linkage, right? So this is called peptide linkage. So what happens here? Here in this NH2, the H from the NH2 and the OH from the acid group here, and one H here, not all the H, and one H here. So goes out. So it forms CONH bond, right? So this is called a peptide bond or peptide linkage, right? So that is also called as an amide. RCONH2 is a formula for amide. You know that, right? RCONH2 RCONH2 is a formula for amide. So here CONH, instead of one hydrogen, this group is replaced. Instead of one R, this group is replaced. Right? So this is also an amide formed between the COH group as well as this NH2 group. Right? So how this what is that amino acids or these proteins are formed? So the proteins are formed by the reaction of amino acids or the joining of amino acids through a peptide linkage, right? So here the number of amino acid involved in this reaction is 2. So we can call the peptide as a dipeptide. So because of the reason two amino acids are involved, we can call it as a dipeptide. So don't think about the peptide bond here. This is a common mistake we'll make often. That is, if there is one peptide bond, we will call it as a monopeptide. No, no, it's not like that. That peptide linkage or the peptide number indicates the number of amino acids involved in that, right? So if two amino acids are involved, it is called as a dipeptide, right? This is a dipeptide, right? And here it is called as glyzalanine or glyzalanine, right? And if one more amino acid is involved in that, if the third amino acid combines, so it is called as a tripeptide. If one more amino acid, fourth amino acid, it contains as a tetrapeptide, right? So similarly, when four, five, or six amino acids are there, it is called as tetra, penta, or hexapeptide. So that is based on the number of amino acids, we can give the name of that peptide. Clear? If more number of amino acids are joined, it is called as a polypeptide, right? Polypeptide or what is that? It's a protein. It is called as a protein and it has more than 1000 micron. The molecular mass of that polypeptide chain will be more than 1000 micron, right? So that can be called as a polypeptide chain. Whether it should be called as a polypeptide chain, that is, which is having molecular mass more than 1000 or amino acid. So there is no clear-cut definition regarding this one, right? So the what is that? அது ஒரு மெல்லிய கோடுன்ற மாதிரி there is no clear-cut definition. It's not very sharp. So both are almost equal. That is the thing we should understand, right? And the polypeptides with fewer amino acids that are also considered as a proteins, right? For example, insulin. Insulin have only 51 amino acids. Insulin have only 51 amino acids, even though it is called as a protein, right? Here, now we had came from amino acid, we had ended in protein. Is that clear? I'll come again one more time. That is, amino acids are joined together. Amino acids are joined together by a linkage called peptide linkage. The peptide linkage is nothing but the acid group of one amino acid. When it reacts with the amine group of another amino acid, a water molecule gets eliminated. There comes a linkage called peptide linkage. It is called as an amide of an acid as well as amine, right? Or amide of an COH group or and NH2 group, right? So this is a peptide linkage, and when two amino acids join together to form a peptide linkage, it is called as a dipeptide. If three amino acid, it is called tripeptide. Four amino acid is called tetra. Like it is gone as a polypeptide. If a polypeptide contains the molecular mass of 1000 micron, ஒரு polypeptideல 1000 micron அளவுக்கு அதனுடைய molecular mass இருந்துச்சுன்னா, we can call it as a protein. So then there comes a thing, what is the difference between a protein and a polypeptide? So it is not very sharp, right? Anyhow, even a polypeptide with 51 amino acids, 51 amino acids. This is also called as a protein, right? So that this is about from amino acid to protein, right? Now let us go for proteins. Proteins that can be classified into two types based on their molecular shape. So fibrous proteins and globular proteins. So from the name itself, we can say fibrous proteins. It look like a fiber, right? It look like a fiber. ஒரு fiber மாதிரி இருக்கும். அதுக்குள்ளார வந்து attachments இருக்கும், right? So that is called fibrous protein. Globular protein will be in spherical shape. Globular protein, it will be what is that complicated structure like this. It will be a complicated structure. So this kind of structure is called globular proteins. Yes, it gives a spherical shape. That is, the chains of polypeptides coils around and gives a spherical shape. Right. These fibrous proteins, we should remember the hydrogen and disulfide bonds. I told you, these are held together. So these are held together by what? These are held together by this hydrogen and disulfide bonds. So then only the fiber-like structure will be formed here. So these proteins are generally insoluble in water, the fibrous proteins, but the globular proteins are soluble in water. An example for fibrous protein, that keratin, because keratin is present in shampoo advertisement, so that we can understand that it's present in hair, right? அப்படிதான் நம்மளுக்கே தெரிய வருது, right? Keratin, that's a fibrous protein. Wool, silk, these are the example of fibrous proteins, and myosin. This is present in the muscle. So these are the example of fibrous proteins. Then if it is globular protein, we can go for albumin. Albumin. You know that they will tell like that. That is, the white of the egg is completely made up of protein. Yes, that's absolutely right. That is albumin here, and insulin. We just now we learned that it consists of 51 amino acids only. So it is considered as a polypeptide or a protein. So what is it again? Or மெல்லிய கோடு. We can call it as a polypeptide as well as a protein. So that are the examples of globular proteins, right? That is fibrous proteins and globular proteins. Is it clear? Right. Then we can go for the structure of proteins, right? So structure of proteins, there may be 3 or 4 structures of protein. That is primary, secondary, tertiary, and quaternary, right? Before understanding that, we can have a look over the picture of these proteins so that it's a symbolical representation, which will give you an idea about how it is, right? Primary structure, that is the arrangement of amino acids in a simplified structure, that is called a primary structure. If the primary structure is coiled, right, that is more number of primary structure if it is joined together, uncoiled, it is called as a secondary structure. And tertiary structure, if more number of secondary structured polypeptide chain are joined together and come to a shape or started much more coiling than the secondary, that is called tertiary structure. And these tertiary proteins or polypeptide chains, when joined together in huge amount, that will form a much more complicated structure, that is called the quaternary structure, right? So these are symbolical representations we can understand easily. Then we'll go for detailed study. Now, so here this is a primary structure, and these primary structure polypeptide chains are joined together to form a secondary structure. And the secondary structures, more number of secondary proteins, secondary structure wala proteins will join together to form a tertiary, and the more number of tertiary wala proteins will join together to form a quaternary structure. Right? So let us get into the deeper thing. Right. Primary structure of proteins. So primary structure in the sense it's a prime, basic, right? Basic thing. So it's nothing but just the sequence of amino acid. So we can remember that it is nothing but the sequence of its amino acids. When the amino acids are joined together, that is called primary structure of proteins. Yes, we know that in each polypeptide chain, the amino acids are linked to each other in a specific sequence, right? That sequence of amino acids or the arrangement of sequence are called this primary structure. If there is any change in the sequence, there will be change in the protein, right? If there is any change in the sequence of arrangement of amino acids, there will be change in the protein, right? Then the secondary structure of protein. Secondary structure is nothing but shape in which along the polypeptide chain can exist, right? என்ன shapeல இருக்கு, either it is in the coiled shape here like this, it's a helix, we call it as a helix, right? So whether if it is in the coiled shape or if it is in the what is it, beta pleated shape. So there are two types of structures, secondary structures of protein. One is alpha helix structure of proteins. Alpha helix structure means there is a axis here. Imagine that there is a axis here. Around the axis, the polypeptide chain of the amino acid has been coiled, right? Has been coiled in right hand side. So that is called as alpha. That is called as alpha helix, right? This is called as alpha helix. And beta pleated structure. Beta pleated structure. Beta pleated structure means there will be hydrogen bonds in between, right? So let us dive into the detailed thing. This structure arises due to the regular folding of the backbone of the polypeptide chain due to the hydrogen bonding, right? So both of these structures arises due to the hydrogen bonding between the C double bond O and the NH groups, right? Of the peptide bond, right? Even though if these groups are bonded together, we already seen here. Look at here, yeah, we had already seen here. That is, one amino acid is joined to another amino acids through the peptide bond, right? So this is a bond between the amino acids. Apart from that, there will be a hydrogen bonding between one amino acid, one peptide and another peptide. Got my point? One peptide and another peptide, right? Yes. Alpha helix is a most common base in a polypeptide chain. It will form hydrogen bonds and twisted itself like a right-handed screw. With NH through NH of each amino acids will make bond with the C double bond of an adjacent turn of helix. So look here. So here there is a, C double bond of group. This makes bonds with the nearby NH group, right? In the turns of helix, it is not like this way, right? It is not like this way. In the Madrikada, it is not in the horizontal way. It is in the vertical way. That is, the carboxyl group in one amino acid makes a hydrogen bonding with the amino group present in another amino acids in the nearby helix, right? So that is called alpha structure, alpha helical structure, right? Is it clear? So listen here, the bonding is not like this. That is, the bonding is between the oxygen and the hydrogen, right? Oxygen and the hydrogen. So oxygen will be from the carboxyl group and hydrogen will be from the amine group. So the in between bond between the carboxyl group and amine group adjacent to each other in the alpha helix, right? So this is called, this is a reason for the alpha helix itself, right? This is the reason for the alpha helix itself. So this is called as the alpha helix structure of proteins, right? Then we can go for beta pleated structure. Beta pleated structure means here this all the polypeptide chain will be what is that? Stretched together, stretched together like something placed on a plain sheet. ஒரு பிளேன் ஷீட்ல எல்லா அமினோ ஆசிடும் பக்கத்து பக்கத்துல ஒன்னோட ஒன்னு அப்படி விரிச்சு வச்ச மாதிரி இருக்கும். ஸோ இங்க பாத்தீங்கன்னா the hydrogen bond will be intermolecular hydrogen bonding. Intermolecular hydrogen bonding. It sends what is that? This is an group, right? This is an amino acid group. There is CH, CO, here N, and here O. This is an amino acid group. This makes hydrogen bonding with another amino acid groups. This makes bonds with another amino acid groups. So this oxygen makes bond with another amino acid group. So it is like this. This hydrogen belongs to this group, right? This hydrogen belongs to this group R CH C double bond O NH, right? So these are called the intermolecular hydrogen. This is the most important point. Remember that the beta pleated structure are due to the intermolecular hydrogen bonds, intermolecular between the molecule, intermolecular hydrogen bonds, and the alpha helix will be due to the intramolecular hydrogen bonds. That's the important question. That is intramolecular, that is intermolecular. So this resembles the pleated folds of a drapery. Therefore, it is called as a beta pleated structure, right? So these are the secondary structures of protein. So secondary structures we have alpha helix and beta pleated structure. Alpha helix is due to the intramolecular hydrogen bonding, and beta pleated structure is due to the intermolecular hydrogen bonding, right? Then we can go for tertiary structure of proteins. So what do you mean by tertiary structure of proteins? This represents the overall folding of the polypeptide chain. So as I shown the picture earlier, that is further folding of the secondary structure, right? So this gives two major molecular shapes, that is called the fibrous and globular, as we discussed earlier. Fibrous proteins and globular proteins that are due to the folding of the more number of tertiary structures, right? This is the tertiary structure of proteins, right? The forces which makes the or which stabilizes the secondary and tertiary structures of proteins are hydrogen bonds, disulfide linkages, van der Waals forces, and electrostatic force of attraction. Mark it as important. So this is an important one mark. The thing which holds the which forces holds the uh, which forces holds the structure of proteins, particularly secondary and tertiary structures. So we can remember hydrogen bonds. We'll know already because that is between the intra and intermolecular hydrogen bonding, that is called beta, and intramolecular hydrogen bonding, that is called alpha, right? So apart from the disulfide linkages, van der Waals forces, and electrostatic force of attraction, right? So then we can go for quaternary structures. Quaternary structure, as we know, it is a much more complicated thing than the tertiary structure, right? And some proteins are composed of two or more polypeptide chains that can be referred as a subunits. Two or more different polypeptide chains. So two or more different polypeptide chains, that is called subunits here. The spatial arrangement of these subunits, how it is arranged with respect to each other, that is called quaternary structure. So what is quaternary structure? If a protein contains more than two or more polypeptide units, polypeptide chains, that we call subunits. If the subunits, how the subunits are arranged with respect to the spatial arrangement, that is called the quaternary structure of proteins, right? So I think you can understand the concept, right? Have a look over the thing in your book and text me the doubts, whatever you are having. We'll clarify then and there. Right? Thank you all. See you next video.